Hey biology students! In this video series, we're going to talk about biotechnology, which is a very diverse and exciting field in biology. In this first video, we're going to discuss genetic engineering, and we're going to talk about one of the very first pharmaceutical products that was made using the techniques of genetic engineering. Many of you may know somebody who takes insulin medication. Why would someone take insulin? People who take insulin have a condition called type 1 or juvenile onset diabetes. What that means is it's a condition that's usually identified in childhood where the person's pancreas does not make insulin. Insulin is a hormone. It's a very important hormone, and it regulates blood glucose levels. So when you eat a sugary meal, a carbohydrate-rich meal, that carbohydrate gets broken down into glucose. Well, we all know the importance of glucose because it's a starting substrate of cellular respiration. But in order to start cellular respiration, glucose has to get inside of your cells into the cytoplasm to initiate glycolysis. Remember all that fun? So how does it get in? Well, it gets in with the help of insulin. So if you don't have insulin, what happens? Well, glucose levels get toxically high in your blood. And this can damage your organs, it can lead to blindness, and have other detrimental effects as well. So people who have this condition, type 1 diabetes, they need to take regular injections of insulin. And what they usually do, they test their blood after a meal. They test the blood with just a prick of the finger, and then they have a machine called a glucometer that measures the amount of glucose in their blood, and they know how much insulin they should be given. So I wanted to show you this medication. It's called Humulin N. It's manufactured by a pharmaceutical company that's based in the San Francisco area, and it's marketed by the pharmaceutical company called Eli Lilly. Let's take a look though at the package. Do you notice something? Let's circle it. On the package, it refers to this human insulin medication as having a recombinant DNA origin. And that's because the technology of genetic engineering sometimes is also referred to as recombinant DNA technology. Notice that that word recombinant has the word combine in it. And that's a really important thing to notice because the big picture of what this technology is doing, it's going to combine DNA from two different species. And those species, in the case of insulin production, are humans. So there's human DNA that's involved in the technology, and then there's also bacterial DNA involved in the technology. This is a really cool story. A lot of people don't know it, although I suppose many of you know somebody who takes insulin medication, and maybe even that person doesn't know how that medication is generated, that it's a genetically engineered medication. Back in the old days, before genetic engineering, insulin was expensive. And that's because they had to take animal pancreas, usually pigs, and grind it up and extract the insulin protein, or from cadavers, people who had donated their bodies to science, grinding up their pancreas to extract the insulin. And that was an expensive process. Nowadays, because we use bacterial cells as the workhorse to do this process, it is a relatively cheap and easy process. So I thought we would start by watching a short animation of how this works, and then I will talk you through the steps.
Recombinant DNA is DNA from different sources, usually different species, combined together in the laboratory for widespread uses in clinical and scientific research. Recombinant DNA usually consists of a gene of interest, here insulin, from a donor organism inserted into a vector, self-replicating DNA from another organism, such as a virus or a plasmid, a small circular piece of bacterial DNA. The DNA from each source is first cut by restriction endonucleases, enzymes that cleave DNA at a particular sequence. The donor DNA is joined to the ends of the vector DNA with the help of the enzyme DNA ligase. The recombinant DNA is then inserted into a host organism, a bacterium or yeast, where it's replicated, creating many copies of the gene of interest. The gene can then be more easily studied, or, as in the case with insulin, translated into protein using the host's cellular machinery.
Okay, so now I'm going to talk you through the steps you just observed in that animation. So the first step in genetic engineering is to identify a trait of interest and isolate the genes that code for that trait. At this point in the semester, you should understand that DNA is composed of genes, which are just segments of DNA, that act almost like recipes, coding for specific traits, and in particular, proteins. So genes create proteins. Remember, that's the central dogma concept, that DNA is transcribed to mRNA and then translated into a protein product. So in the case of manufacturing insulin, we would say that the gene of interest is the insulin gene, the human insulin gene. So what would you have to do? Well, you would have to take the DNA. So let's draw a DNA molecule here. So we'd have to take a DNA molecule from humans. So we would need a sample of human DNA. This could be from a blood sample. This could be from something even less painful than that, which would be just a cheek swab or a saliva sample, assuming that there's enough DNA or enough cells in there. So that DNA would be extracted. We're interested in cutting out the gene that codes for insulin. But even though it's the pancreas, the cells in the pancreas that make your insulin, you can actually get this gene from any DNA sample, assuming it's from a human who has a functional copy of the insulin gene. And that's because every cell in our body has the same genome, okay? With the exception of your sex cells, but every somatic cell has the exact same DNA. Isn't that interesting? So what happens is it's just the cells in your pancreas that express the insulin gene, meaning it's there that that gene gets switched on, if you will, and then that protein is translated. Anyway, so in this sequence of human DNA, we would first have to isolate where that specific gene for insulin is. And then we would need to actually extract it or cut it out. So let's, for simplicity's sake, that this region right in here, this is where we find the human insulin gene. This is the sequence of human insulin gene. And the first step is to then isolate the gene, right? We need to isolate it. Well, that's because there are these amazing little molecular scissors. I'm going to draw scissors here to the best of my ability here. So these are my little scissors. And these scissors are called restriction enzymes. In the video, they called them restriction endonucleases, but restriction enzyme is fine. So these restriction enzymes, they act like molecular scissors on the DNA molecule. What that means is that these are specific enzymes, and they were actually originally identified in bacteria in like the 1970s. So we've known about them for a long time. And see, bacteria make these enzymes that they use to snip DNA at specific sequences. And the bacteria use them actually as sort of a defense against viruses. But we in biotechnology can harness the ability of using restriction enzymes because you can use that enzyme to cut a specific sequence that you want to remove from the DNA. Okay, so imagine now we're cutting out this human insulin gene, just that gene. We don't want all the rest of the DNA, what codes for all the other human traits. We just want this gene. This is the one. This is the one we want because it's the recipe for making the insulin protein, which is what we need to give to the diabetics. Okay. So we isolate it using the restriction enzymes. And so imagine with me then that this is all within a test tube. So it's really hard to see. That's why we draw it out like this. So imagine now we're going to be cutting out that specific gene. It's going to be cut at sequences on the DNA and by these enzymes, okay, by these little molecular scissor enzymes. So that piece of DNA is going to be removed. Now, if we want to use maybe some color, let's actually just, for simplicity, let's just make the human insulin gene just a red line, if you will. So it is a piece of DNA, but this red line would represent that human insulin gene. Okay, now what are we going to do with it? Well, step 2. Step 2, insert the gene of interest into a desired organism. Okay, so now we have the recipe. Do you get that? So we have the recipe for making insulin. So we need to put it into the organism that we want to make that insulin. And what scientists figured out is that they can take this insulin gene and they can put it into bacteria. And the way that we deliver it into a bacterial cell, we deliver it into the bacterial cell using a piece of circular DNA that you saw described in that video. And that piece of circular DNA is called a plasmid. So this is called a plasmid. And a lot of times we use this term vector when we're talking about how we're going to deliver our gene of interest. So in this plasmid DNA, and this is actually bacterial DNA, and these are relatively small loops of DNA that can be manipulated in a laboratory procedure in a test tube again, where what we can do is we can use restriction enzymes. So let's make some scissors here. So we can treat the enzyme or treat the DNA with the enzymes, and we can get that segment of DNA removed. And then we can mix it. So this is going to be mixed with the human insulin gene, which we were using red for the human insulin gene. So you mix them, and what happens is the scissors, the restriction enzymes, they cut out that particular piece of DNA, and then it allows for the human gene to be sort of glued. It's like a cut and paste sort of thing, because there's actually another type of enzyme. They talk about it in the video. It was called a ligase. Okay, so the scissors, this is the restriction enzymes. We'll just label them RE. This red line, this represents the human insulin gene. And so what happens is you mix this together and voila, we get what we call a recombinant DNA molecule. So in blue, remember in blue, that's bacterial DNA. In red, that's human DNA. That's specifically the human insulin gene that gets inserted in there. And so the insulin gene, think of it as being glued to the bacterial plasmid DNA. And the result is what we call a recombinant DNA molecule. Recombinant, remember, means combine. And it actually is sort of like a hybrid, is another way to think of it, as a hybrid molecule, because in red, that's DNA from the humans, the human gene for insulin there. And then in blue, there's our bacterial DNA. Okay, they've hybridized or glued together in this lab process. So now what happens is we take this gene of interest, so the gene of interest, and we want to deliver it. So the vector, we say, is this plasmid. It's acting as a delivery mechanism because in natural environment, bacteria will take up foreign DNA through a process we call transformation. So this is actually a really easy process to model in a laboratory. We actually do it in microbiology at Palomar. So it's something that can be accomplished in a laboratory setting at our college. So it's really simple to do. You simply just mix bacterial cells in a test tube and with this plasmid, this recombinant DNA plasmid, and a couple steps involved there, but you are able to get these bacteria to swallow up, okay, this hybrid DNA molecule. It's pretty cool. So now let's draw a bacterial cell. So this would be a bacterial cell. So the next step would be through a process that we call transformation. Which is a laboratory process. You don't need to know all the details about it, but basically it's just mixing the bacteria with the gene that you want, the plasmid you want to be delivered. And imagine that bacterial cell sort of swallows up this piece of DNA. Now bacteria will have their own DNA. So we'll say here's the bacterial DNA because it's a functional bacterial cell. But then what you're giving them is you're giving them this extra piece of DNA. So they pick up this extra piece of DNA and it enters their cytoplasm. So let's draw it in here. Okay, so now this bacterial cell has the plasmid. It has the instructions for making human insulin. Now, here's another important thing you need to remember from the previous lectures on molecular genetics. Do you remember there is a very important feature of the genetic code that's going to make what you're about to see possible? And that feature is that the genetic code of life is universal. Do you remember what that means? It means that the DNA from one organism can be transcribed and translated in the same way across all life on Earth. Why? Well, because we're all related. We're related in an evolutionary chain of connection going all the way back to the very first cells. All cells on Earth have the same basic steps for creating proteins from our genes. And the amazing thing is that this bacterial cell takes that human insulin gene and just says, okay, here's a recipe. This recipe, it looks just the same as all my other recipes, and I'm going to transcribe it and I'm going to translate it into a protein. And it turns out that the protein that's made from this bacterial cell is identical to the protein made from human cells. Let's diagram that out in the next slide. So this is going to be step 3. Step 3 is that the organism that has now been genetically engineered. So we just genetically engineered that bacterial cell. It's going to grow in the lab. It's going to replicate itself, but it now has a new trait. And what's the new trait? The ability to make human insulin. That is pretty cool. So now imagine that we have duplicated, because basically you just let the bacteria grow, give them food, and they just copy themselves. So we get, this is going to be just billions. So you can get billions of bacterial cells relatively quickly, even just within a few hours, because they grow exponentially. And all of them, so we'll say they have their regular DNA. Basically, we're going to recopy what I had on the other slide. Hold on. So now we have some representative duplicates of the same type of cell here, because the cells have divided by their binary fission process, their exponential growth process, to be identical to each other. And now they all carry this recombinant DNA plasmid, and now they all have the ability to make or translate the human insulin protein. So let's go ahead and show that there's human insulin protein being made. We'll make it these little red dots. So I'll get all these little red dots going here. And that would be translated protein. So this would be let's write this, human insulin protein. Do you see this now? Do you see that bacteria are actually making a human protein? And the reason why that's possible is because we are all related. We are related even to bacteria. Bacterial enzymes are really no different than the enzymes you have in your cells that are making your proteins. So you give them a recipe for making insulin, it's just a different kitchen. It's just like if you are used to making food in your kitchen, but then you go over to somebody else's house and you make food, the same food, just in a different kitchen. That is analogous to this situation. It's the same recipe as what your cells are doing, but it's in the bacterial cell, using the bacterial enzymes and the bacterial ribosomes to make the human insulin. And so what happens is that the protein is extracted. So then the next step would be actually to isolate the human insulin protein from the bacteria. Sometimes I have students that get confused about this, and they tell me on an exam, because I always ask about this, they tell me, oh yeah, we're giving the diabetic bacteria, and that's not what's happening, okay? So let's just clarify that real quick, is that we're using the bacteria to make the protein, okay? Because the bacteria will translate it using the ribosomes in the exact same way that we studied the central dogma. So the central dogma is universal. So bacteria will transcribe a gene to mRNA, and bacteria will translate the mRNA into a protein using bacterial ribosomes, okay? But we don't give the patients, our diabetic patients, we don't give them the bacteria, okay? What we do is we then lyse the cells. What that means is to basically break open all the bacteria. So you just mix them in another, test tube, mix them, and you use chemicals that break open the cells. And so you break open these cells, and then you release all of this protein and purify it. You want to get it away from any bacterial proteins. You want to make sure that it's chemically identical to what we would give our patients. So this is going to be a test tube of the insulin medication. So what's in there? Well, we were making insulin red dots here to represent this is insulin protein. So understand that that is the end product of this process. That protein is isolated. We isolate it. Just again, like the analogy, the bacteria is basically just like a kitchen where you're cooking up the insulin protein. But of course, once you cook up that particular cake, right? You actually eat the cake, you don't eat the kitchen, right? So we're not giving them the bacterial cells, we're giving them the product of what the bacteria made, which was insulin. And it's made from bacteria. So that's why you see the term that this is recombinant. It's recombinant insulin made from the process of genetic engineering. And I hope that was interesting for you, and we'll talk more biotechnology in the next videos.